everyone. In this little video, I'll be showing you how the negative transcriptional regulation works. So negative transcriptional regulation uses a protein called the repressor. The repressor binds to the operator on the DNA. The operator is a regulatory DNA element that is downstream of the promoter, but upstream of the coding part of the DNA that's going to be used to make the transcript. So what happens when the repressor is bound, the sigma factor and RNA polymerase either cannot attach to the promoter at all, or they can attach but they can't move down and transcribe because they're physically blocked. So when the repressor is attached to the operator, there's no transcription happening. Okay? So we know that there are two basic types of genes: inducible genes and repressible genes. So the inducible genes are going to be the ones that are involved in catabolic processes. So these genes code for proteins that we need to use to break down nutrients. And so these genes are not going to be normally expressed, but when the bacterium encounters a new nutrient, we will need to upregulate these genes. All right? So the inducible genes, the normal state for them is with the repressor present on the operator, such that these genes are not expressed and there is no transcription happening. When the bacterium encounters the nutrient that goes with this protein that's encoded by this gene, then either the nutrient or a metabolic product of the nutrient, which is going to be the inducer in this case, comes and binds to the repressor on the DNA. The binding of the inducer, which you'll remember is also called generally an effector molecule, changes the shape of the repressor, because we know that when effector molecules bind to proteins, they change the shape. So the inducer binds to the repressor changes the shape and detaches from the DNA and goes into the cytoplasm. Now, the sigma factor, which I'm not going to show you, and the RNA polymerase can attach to the promoter, move down the DNA, and transcribe the required messenger RNA. All right? The second type of gene that we talked about is the repressible gene. So repressible genes are going to be those that are involved in anabolic processes. We are going to normally be expressing these genes until we don't need the product of these genes anymore, and then the gene expression will be downregulated. So for repressible genes, the repressor is going to start off in the weird shape here in the cytoplasm, the not binding to the DNA shape. When we have an indication that we no longer need the proteins that this gene is producing, then we'll have a co-repressor. Another small molecule, also generally called an effector, is going to come and bind to the repressor in the cytoplasm. Okay? When the co-repressor binds to the repressor, it's going to change the repressor back to the DNA binding shape. The repressor is then going to bind to the operator, preventing RNA polymerase from binding and moving down, and thereby stopping expression of these genes.